Hello, my dear students. Today I will discuss on the topic reproduction in fungi. So let's start. First of all, fungi can reproduce by asexual, sexual, and vegetative means. In vegetative reproduction, a part of mycelium separate and forms a new individual. And various methods of vegetative reproduction are fragmentation, fission, and budding. What is fragmentation? In case of fragmentation, the hyphae break into smaller fragments, and then each fragment, upon getting suitable condition or favorable condition, germinate to form a new mycelium. Then fission, in this method, splitting of the cell occur to form two daughter cells. It occurs by the formation of constriction between two cells. Then budding, a small bud formed from parent cell which gradually increases in size and receive a part of nucleus. A cell wall is formed which separates the daughter cell from the parent cell. So these are two main methods of vegetative reproduction seen in case of fungi. Then by sclerosia, what is sclerosia? These are parenating bodies formed by the compact masses of interwoven hyphae. Sclerosia under suitable conditions germinate to form new individual, as seen in case of claviceps and sclerotinia. Sclerotinia is a parenating body, that means in unfavorable condition, it will undergo a dormant condition. Then again, when the favorable condition will come, it will germinate and form a new individual. Then rhizophyte. It is a root-like structure. As the name suggests already, rhizo means root-like. So rhizophyte is a root-like structure, root-like elongated mycelial strand. They remain dormant under unfavorable condition, and on coming of the favorable condition, it will develop a new mycelium again. Then asexual reproduction, it is commonly takes place through spores, either motile spore or non-motile spore, and form a specialized part of mycelium. The various types of spores are zoospore. These are commonly found in lower group of fungi. For example, saprolegnia and pythium. They are equipped with one or two flagella. That means they are motile. Zoospores are motile because they have flagella. Then sporangiospore. Sporangiospore or aplanospores are non-motile. They lack flagella and formed inside a sporangium. That means inside a sac-like structure called sporangium. For example, mucor and rhizospores. These spores may be uninucleate or multinucleate and possess two-layered cell wall. Sporangiospore have two-layered cell wall. Then conidia. They they are produced externally on a branch or unbranched hyphal tip termed as conidiophores. The conidia may be singly or in chain. Conidia may be uninucleate or multinucleate. The multinucleate is more common in the members of Deuteromycetes. Then oidea, they are formed by fragmentation of hyphae. Hyphae gets fragmented and each fragment form a form as a separate spore, germinate to form a new mycelium. This is the case of oidea. Then chlamydospore, they are formed by rounding off and enlargement of terminal or intercalary cells of hyphae. These can be single or formed in chain. They do not separate from hyphae but remain viable and germinate under favorable conditions. Then ascospore. Ascospore is produced inside a ascus. Then this kind of spore is specific to fungi Ascomycetes. Ascomycetes formed form ascus, and inside ascus, ascospores are formed. Then uridospore, these are thin-walled red summer spore of a rust fungi produced usually on leaves or stem of grasses. Then basidiospore, these spores are produced by Basidiomycetes fungi. They are typically contain one haploid nucleus that is the product of meiosis, and they are produced by specialized fungal cell called basidia. So basidiospore is formed inside basidia in case of basidiomycetes fungi. Then sexual reproduction. It involves the formation of fusion of gametes, but in case of asexual reproduction, it lacks the involvement of two gametes. It is uniparental, but sexual reproduction is biparental, involves two gametes for fusion. Sexual reproduction found in all groups of fungi except deuteromycetes. So, deuteromycetes is known as fungi imperfecti. Then, sexual reproduction has three distinct phases. What are those phases? The first phase is plasmogamy or protoplasmic fusion. Here only the protoplasmic content gets fused. Then second phase, karyogamy, that means fusion of nuclei, and third phase meiosis, which is the reduction division of zygote. In first phase, the protoplasmic contents gets fused. Then in second phase, karyogamy occur, that means fusion of two nuclei. Then meiosis occur. After the fusion of two nuclei, one zygote is formed. Then the reduction division of that zygote occur. Then the various methods of sexual reproduction in fungi are as follows. One is planogametic copulation. This is the simplest type of sexual reproduction. In this process, fusion of two gametes of opposite sex or strain takes place, whereas one or both fusing gametes are motile. Here both are motile. It results in formation of diploid zygote. Two gametes fuse to form one diploid zygote. This process is usually of three types. One is isogamy, where the fusing gametes are morphologically similar. That means they look similar, but and motile, but physiologically dissimilar. These gametes are produced by different parents. For example, in syncytium. Then heterogamy, where the fusing gametes are morphologically as well as physiologically different. So they are known as heterogamy. Heterogamy reproduction is of two types. One is anisogamy, another one is oogamy. Next, in case of anisogamy, this fusion of two motile gametes occur where the male gamete is small and more active than female gamete. For example, allomyces. But in case of oogamy, the female gamete is totally non-motile, but the male gamete is motile and fuses with large non-motile female gamete. In case of anisogamy, the female gamete is less active than male gamete, but in case of oogamy, male gametes are only motile, but female gametes are non-motile. Male gametes are small in size as compared to your female gametes. Then gametangial contact. It is a process of sexual reproduction. In this process, two gametangia of opposite sex or opposite strain come in contact with one another. Then the male gametangium transfer male nucleus or gamete into female gametangium, either through pore at the point of contact or through fertilization tube. For example, Phytophthora, albugo and pythium. Then gametangial copulation. It involves fusion of entire content of gametangia to form common cell called zygote or zygospore, as seen in case of mucor and rhizopus. So what is the difference between the previous one and this gametangial copulation? In the previous one, it involves only the fusion of nucleus or transfer of nucleus, but here it is the fusion of entire content of gametangia. Then spermatization. The fungi produce many minute spore-like single-cell structure called spermatia or spermatia, which are non-motile gametes. These structures are transferred through agency of water, wind, insect to either a special receptive hyphae or trichogyny of ascogonium. The content migrate into the receptive structure. Thus, eukaryotic condition is established as seen in case of Poxinia. Then somatogamy. This takes place in fungi where formation of gamete is absent. In such fungi, anastomosis takes place between hyphae and their somatic cell. They fuse to produce dikaryotic cell. Here two vegetative cells fuse to form dikaryotic cell, as seen in case of Agaricus, Peniophora, etc. So these are all about different modes of reproduction in fungi. In my next class, I will discuss about the economic importance of fungi. So stay tuned and enjoy my video. Thank you.